The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
So let me start by first, I already alluded to this, but let's talk about the problem of vision. So, we all look at, this is just one computational challenge that our brains solve, but it's one that many of us work in, are very fascinated by. But as you'll hear in the rest of the course, there are other problems that are equally fascinating. But I'm going to talk about problems of vision, and I'm going to talk about a specific problem of vision, and that's the problem of object recognition. So I will try to operationalize that for you. And one thing you'll see when I talk is that our field, even though we can be motivated by words like vision and object recognition, we can only make progress if we start to operationally define things and then decide what domain models are going to apply. And I think that's an important lesson that I hope will come across in my talk. So this is the way computer vision operationally defines part of the problem of object recognition and vision. It's as if you take a scene like this and you want to do things like come up with an answer space that looks like this, where you have noun labels, say car, and you have what are called bounding boxes around the cars. Similarly for people or buildings or trees or whatever nouns that you or DARPA or whoever wants to actually label, right? So this is just one way of operationalizing vision, but I think it gets at the crux of what we're after, which is there is latent content, what's called latent content in this image, that all of us instantly sort of bring to our memories, that we can say, aha, that's a car, that's a building. They're nouns that pop into our heads. We also know other latent information about these things, like the pose of this car, the position of the car, the size of the car. The key point that I'm going to tell you today about this problem is that that information feels to us that it's sort of obvious, but it's quite latent in the image. That's implicit in the pixel representation. Those of you who have worked on this problem will understand this, and those of you who haven't, hopefully I'll give you some flavor for what that problem feels like. So I want to sort of back up a bit, and this is more from a cognitive science perspective or a human brain perspective, to ask, why would we even bother worrying about this problem of object recognition? And maybe this is obvious that those of you, I don't need to say this, but I like to point out that we think of the representations of the tokens of what's out there in the world as being the substrates of what you might do what's called higher level cognition. Things like memory, value judgments, decisions and actions in the world. You can't imagine building a robot and having it try to act in the world and it doesn't even really know what's out there. So these are the sort of substrate of these kind of cognitive processes. Again, from an engineering perspective, these are kind of processes or sort of behaviors. This is just a short list of them that might depend on your good abilities to recognize and discriminate among different objects. And I think if you look through this list, you could imagine things that would go terribly wrong if you didn't actually do a good job at identifying what's out there in the world. Okay, so that's just to think about, again, as an engineer building a robot. I want to kind of connect, this is a slide I stuck in, and I want to connect to this course the idea that I know many of you are from maybe these backgrounds or from this background. And when I think about the brain, I have this coin here to say, really, these are kind of two sides. We're studying the same coin from two directions here. And really the question that we have to all be excited about, I hope many of you are excited about, is how does the brain work? You could do computer science and not care at all about this question. I think it's a little harder to do these and not care about this question, but it's possible, I guess. But so these are all trying to answer this question. And then, I mean, this is maybe pretty obvious, but when you have biological brains that are performing tasks better than current computer systems, machines that humans have built, then the flow tends to kind of want to go this way. You kind of discover phenomena or constraints over here. These lead to ideas that can be built into computer code that can say, hey, can I build a better machine based on what we discover over here? And many of us came into the field excited to do this and are still excited of this kind of direction. But an equally important direction is that when you have systems that are matched with our abilities or that can compute some of the things that we think the brain has to compute, then the flow goes more this way, where there's many possible ways to implement an idea, and these become falsifiable. That is, that they can be tested against experimental data to ask which of these many ways of implementing a computation are the ones that are actually occurring in the brain. And that's important if you say you want to build brain-machine interfaces or fix diseases or do something that's on the level of interacting with the brain directly. So I hope that you guys keep this picture in mind, because I think it's sort of the spirit of the course that both of these directions are important, and it's not as if we work on this for 20 years and then work on this for 20 years. It's really the flow across them that I think is most exciting to us. So just to connect to that, a little bit of history is sort of where was the field on this problem of visual recognition. I don't know if many of you heard this, but here you are at summer school. So there was a summer vision project, it was called, at MIT. I used to think this story was apocryphal. In 1966, there was a project that the final goal was object identification, which will actually name objects by matching the vocabulary of known objects. So this was essentially a summer project to say, we're going to get a couple undergraduate students together and we're going to build a recognition system in 1966. And this is sort of the excitement of AI. We can build anything that we want. And of course, those of you who know this problem turned out to be much, much harder than anticipated, right? So sometimes problems that seem easy for us are actually quite difficult. If any of you wants this, I'm happy to share this document with you. It's sort of interesting, the space of objects that they describe, things like recognizing, of course, they'll say like coffee cups on your desk, but they'll also say packs of cigarettes on your desk, right? So this sort of dates the time of this here, right? So it's a little bit like Mad Men or something. So now here we are today, and I guess I just can't help but sort of get excited about, here's this like really cool machine that's just amazing that does these computations. The thing's got, I can't tell you all this because 100 billion computing elements, solves problems not solvable by any previous machine, and the thing, it looks crazy, but it only requires 20 watts of power. And the key here is, and those of you who've seen this slide, I'm not talking about this thing, I'm talking about that thing right there, right? So this is sort of a scale of what we're after. And we often don't talk about power, but this is something engineers are especially interested in as they build these systems, is how does our brain solve these problems at such a low wattage, so to speak, right? So this is, again, the spirit of many of the things that I hope that you guys are excited about in the future of this field. Here's another slide that I pulled out that I often like to show, is that from an engineer's point of view, we often try to say, well, we want to build machines that are as good as or better than our brains. So machines today, you guys know this, beat us at many things. Straight calculation, they beat us at chess. When I was a grad student, they recently won at Jeopardy. In memory, they've always beaten us. I mean, machines are way better memory than us in the simple form of memory. Seeing in pattern matching, go to the grocery store, hey, what's that barcode done? I don't know what that was, but it just scans in and somehow it does pattern matching, right? So there's forms of vision that machines are way better than us. But some forms of vision that are more complicated, that require generalization, like object recognition or more broadly scene understanding, we like to think that we are still the winners at, and even things that we take for granted, like walking. This is quite a challenging problem. Now, engineers really want to sort of move this over here, right? So our goal is to discover how the brain solves object recognition. And the reason I put this up is from an engineering point of view, that just doesn't mean write a bunch of papers in a textbook that says this part of the brain does it, but actually help to implement a system where this is at least matched with us, and perhaps, and I assume someday will be better than us. And this is also a problem that it's a gateway problem. That is, even if it's just this domain, we think that the systems we're studying might generalize to other, for instance, sensory domains. And I think you'll hear this. Gabrielle told me you were going to do an auditory visual comparison session later in the week. OK, that's an engineer's point of view. How do I just build better systems? Let's step back and talk from a scientist's point of view. So this is really now to introduce the talk that I'm going to give you today. So when you're a scientist, what's our job? We say we want to understand. We all write that, understand, right? What does that mean? Well, what it really means, if you boil it down, and I would love to discuss this if you like, is that you have some measurements in subdomain. So you can think of this as a state space here. We're like, this is like the position of the planets today, and this is like the position of the planets tomorrow, right? So, or you could say this is the DNA sequence inside a cell, and this is some protein that's going to get made. So you're searching for mappings that are predictive from one domain to another. And we could give lots of examples of what we call successful science where that's true. Is being able, this is the core of science, is to predict, given some measurements or observations, what's going to happen either in the future or some other set of measurements. So predictive power is the core of all science and the core of understanding. And I think it'd be fun if you want to debate that you think there's another way, but this is what I come to in thinking about this problem. And the accuracy, the reason I'm bringing this up is because the accuracy of this predictive mapping is a measure of the strength of any scientific field. And some fields are further along than others. And I would say ours is still not very far along. And our job is to bring it from a non-predictive state to a very predictive state. And so that means building models that can be falsified and that can predict things. And you'll hear that through my talk. As Gabrielle mentioned, what we try to do is build models that can predict either behavior or neural activity. And that's what we think is what progress looks like. So now let's translate this to the problem I gave you, which is the problem of vision, or more generally object recognition. You could imagine there's a domain of images. So just to slow down here, just so everybody's on the same page, Each dot here might be all the pixels in this image, and this dot all the pixels in this image. So there's a set of possible pixel images that you could see. And we imagine that they give rise to, in the brain, some state space. Think of this as the whole brain for now to keep it sort of just fix ideas. That you could imagine that this image, when you're looking at it, gives rise to some pattern of activity across your whole brain. And this image gives rise to a different pattern of activity across your whole brain. And loosely, we call this the neural representation of this thing. But then what we do is somehow, when we ask you for behavioral reports, there's a mapping between that neural state space and what we measure as the output. Whether you say it or write it, you might say that's a face. These are both faces if I asked you for nouns among them. Okay, so this is another domain of measurement. Okay, so now you can see I'm setting up the notion of predictivity. And what we want to do is we have this complex thing over here of images that somehow map internally into neural activity and then somehow map to the thing we call perceptual reports. And notice I've already put things that we call nouns that we usually associate with objects: cars, face, dogs, cats, clocks, and so forth. Okay, so understanding this mapping in a predictive sense is really a summary of what our part of the field is about. And again, accurate predictivity is the core product of the science. That underlies our ability to build a system like this. Many of you are interested to fix a system like this, or to perhaps even augment our own systems. If we want to inject signals here and have them give rise to percepts, we have to know how this works. A big part of the field of vision has spent a lot of the last, say, 3 decades working on the mapping between images and neural activity. That's usually called encoding, predictive encoding mechanisms, and it's just driven by Hubel and Wiesel's work. The people saw this as a great way forward. It's like, let's go study the neurons and try to understand what in the image is driving them. What's an image computable model in the ideal world that would go from images to neural responses. The other part is that there's some linkage, we think, between the neural activity and these reports. And notice this is actually why most of us get into neuroscience, because you notice this arrow is two-way. This is actually quite deep here. From an engineer's point of view, you go, well, there's got to be some mapping between the neural activity and the button presses on my fingers or my saying the word noun. There's some causal linkage between this and the things that we observe objectively in a subject. But this is where philosophers debate about, like, well, in some sense, these are sort of two sides of the same coin. We say our own perception. There's some aspects of the internal activity that are the thing that we call awareness or perception. Now, I'm not going to get into all that, but I just want to point out that if you're just building models, you can't approach that. It's this strange thing between neurons and these reported states that many of us are fascinated by. So this is called predictive decoding mechanisms. For me, it's all going to be operationalized in terms of reports from humans or animals. And I'll not do that philosophical part, but I thought I'd mention that for those of you who like to think about those things. So for visual object perception, I want to point out that, again, the history of the field has been mostly here. This link has been neglected or dominated by weakly predictive word models. That doesn't mean they're not useful starting points, but they're weakly predictive. And so a weakly predictive word model would be infertemporal cortex, a part of the brain I'm going to tell you about today, does object recognition. That model has been around for a long time. It is somewhat predictive because it says you take that out and all object recognition would get destroyed, would be a prediction. Turns out that doesn't actually happen. We can discuss that. But it doesn't sort of tell you how it does it, how to inject signals, which tasks are more or less affected, right? So that's what I mean by weakly predictive. It's a word model. Face neurons do face tasks. That's probably true to some extent, but again, it doesn't tell us. It's more tight. It sort of says, oh, I'll take out these smaller regions and there'll be some set of tasks that sort of involve faces. I don't know. I won't say anything about other tasks. So that's a somewhat more strongly predictive model, but still pretty weakly predictive. And my personal favorite that comes in from reviewers a lot is attention solves that, right? So this is just a statement that just to be on the lookout for word models that don't actually have content in terms of prediction, right? I don't know what that means. I read this as hand of God reaches in and solves the problem, right? So there's got to be an actual predictive model that can be falsified. Okay, so I don't mean to sort of doubt the importance of these before people start giving me a hard time. There are attentional phenomena. There are face neurons. There is an IT. That's what we study. I'm just trying to emphasize for you that we need to go beyond word models into actual testable models that make predictions that would stand even if the person claiming those models is no longer around. It would make a prediction. Let me try to define a domain. I said we're going to try to define stuff. It's hard to define stuff. It's big. Vision, it's a big area. Object recognition, I sort of said it vaguely, and when I say this, I include faces as an object, a socially important one. You'll hear this from Vinrik, I think. But I want to kind of say, to try to limit it even further, that's still a big domain. And so we tried early on to reduce the problem even further to something that is more, again, naturalistic, that we think can give us more traction in this predictive sense. So we started by saying when you take a scene like this and you analyze it, you may not notice it, but your ventral stream, or really your retina, has high acuity in, say, the central 10 degrees. There's anatomy that I'll show you later that the ventral stream is especially interested in processing the central 10 degrees of information. So that's about 2 hands at arm's length, those of you in the room. So you may have the sense that you know what's out there, but you don't really, right? You kind of stitch that together. And the way lots of people have shown this, the way you stitch this together is making by rapid eye movements around called saccades, followed by fixations, which are 200 to 500 milliseconds in duration. You don't really see during this time here. It's not as if your brain shuts down. It's just that the movement's too fast for your retina to really keep up with this. So you make these rapid eye movements. You fixate, fixate, fixate. And what you do is that brings this sort of sampled scene to the central 10 degrees that might look something like this. So those are 200 millisecond snapshots across that scan path. And I'll play for you one more time. Now you should notice that there's one or more objects in each and every image that you probably kind of said, oh, there's a sign, there's a person, there's a car. You might have gotten 2 out of each one, but you were sort of extracting, at least intuitively to me, at least 1 or more kind of foreground or central objects when I show you those images. And that ability to do what I just showed you there, we think is the core of how you analyze or build up a scene like this, at least how the ventral stream contributes. And therefore we call that core recognition, which I define as a central 10 degrees of visual field, 100 to 200 millisecond viewing duration. And again, it's not all of object recognition, but we think it's sort of a good starting point. And the way that we partly got into this is because of rapid serial visual presentation movies from the 70s. Molly Potter showed this really nicely. This is a movie that I've been showing for 15 years now. Notice that this is just a sequence of images where there's typically 1 or more foreground objects, and you should be quickly mapping those to memory, even though I'm not telling you what to expect. Like Leaning Tower of Pisa, right? I'm not going to tell you that you're going to see Star Wars characters, well, I just did, but you quickly are able to map those things to some noun or even a more precise subordinate noun, and know this is Yoda. Right? And so our ability to do that, we're very, very good at that. Notice you didn't need a lot of pre-cueing, yet you're still able to do that. And that is really what fascinates us about vision or object recognition in particular. Even without featural attention or pre-cueing, you're able to do a remarkable amount of processing. And I think that's a great demonstration of that. And just to quantify this for you, because sometimes people say, well, you're showing it too short, your visual system doesn't do much. Here's an 8-way categorization task. I'll show you later under a range of transformation. These are just the example images of 8 different categories of objects. It doesn't really matter what I might do here. You get a very similar curve, and that is you get most of the performance gain in about the first 100 milliseconds. This is accuracy, about 85% correct. This is a challenging task, as I'll show you earlier. It looks easy here, but it's quite challenging. 85% correct. If I let you look at the image longer, up to 2 seconds, you can bump up to around 90s. So there is some gain with longer viewing duration, but you get, chances 50. So you get this huge ability and we're not the first to show this. This is just to show you in our own kind of task. That the data I'm going to tell you about, where we show the image for 100 to 200 milliseconds, this is the typical primate viewing duration that I pin this on. We use this for reasons of just efficiency. But you see the performance is similar across that time. You get a lot done. Your visual system does a lot of work in that first glimpse. And that's core recognition that we are trying to study here. And I know it's not all of object recognition or all of vision, but it's now, we think, a much more defined domain that we can make progress on. And that's what we've been working on, and that's essentially what I'm going to talk about today. So think of vision, object recognition, within that core recognition. This is David Marr. And David and actually Tommy Poggio, a long time, and Tommy wrote the introduction to David's. If you guys haven't read this book, Vision, does anybody of you guys know this book? It's really a classic book in our field. It's the first couple chapters that are the part you should really read. I mean, that's the best part of the book. And one of the things that you take from this book that I think David and Tommy helped to lay out a long time ago is that there's this challenge of level, that you don't come into a problem. Like, I think one of the things I take from this is now they would try to define 3 clean levels. It turns out not to be this clean in practice, but there's one level called computational theory. What's the goal? What's appropriate? What's the logic? And by what strategy in which it can be carried out. There's another level which is, okay, now once you decide that, how should you represent the data? How can you implement an algorithm to do it? And then there's this actually, how do you run it? How do you build it in hardware? And neuroscientists often come in, they're like, I'm going to study neurons, and it's sort of like jumping into your iPhone and saying, I'm going to study transistors, right? They often tend to sort of start at the hardware level. And I think that's sort of the biggest lesson taken. It's like, oh, there's something going on here. Those transistors are flying, and make some story about it if you were recording from the brain or measuring transistors in my iPhone. But I think the important point to take from this is it helps to start thinking about what's the point of the system, what might it be doing, how might you solve that problem, and that leads you then to algorithm, and then you think about representation. So it's sort of a top-down approach rather than just digging into the brain and sort of hoping that the answers will emerge. So I'm going to try to kind of give you that sort of top-down approach in this problem that I'm talking about. I've already given you a bit of it by introducing you to the problem. I'll say a little bit more about that and step down a little bit this way. And so this kind of a thinking, I think, is important to making progress in how the brain computes things. So here's a related slide that I made a long time ago that, again, I pulled out for you guys that I think helps bridge between what I just said about the MAR levels of analysis and whether you're a neuroscientist or cognitive scientist and or a computer vision or machine learning person. So the first is, what is the problem we're trying to solve? So that's the MAR computational level 1. So computational vision, so now operationally, you'll hear folks in machine learning, they might say, well, there's some benchmarks, that's good. There's like a ImageNet challenge or whatever challenge they want to solve. Sometimes they'll say, well, the brain solves it. That's not good because they didn't really define the problem. Neuroscientists will say, well, it's something like perception or behavior, or there's some sort of behavior that they imagined, although characterizing that behavior is not usually their primary goal, but I think there's at least some progress in that regard. Now there's like, what does a solution look like? So this is really just to talk about language. So useful image representations for machine learning, like what we might call features. But neuroscientists will talk about explicit neuronal spiking populations. You heard this in Haim's talk a bit. He was sort of using these words interchangeably. Again, this may be sort of obvious to you guys, but I thought it's worth going through. So this is like MAR level 2, like representation. How do we instantiate these solutions? So this is still kind of level 2, algorithms or mechanisms that actually build useful feature representations. Neuroscientists will think about neuronal wiring and weighting patterns that are actually executing those algorithms, right? This is what we think is a sort of a bridging language there. And then there's this deeper level that came up in the questions, which is really, how would you construct it kind of from the beginning, right? Learning rules, initial conditions, training images are words that are used here. There's sort of a learning machine. Here, for neuroscientists, talk about plasticity, architecture, and experience, right? So, but again, those are sort of similar questions, just with different language. And I'm doing this because I think the spirit of this course is to try to build these links kind of at all these different levels here. Okay, so hopefully that kind of helps orient you to how we think about it. Let me just go and say, I want to talk about number 1. What is the problem we're trying to solve? And why is it hard? So I said object recognition is hard, and I showed you that MIT challenge, and it was difficult. Maybe it's hard because there's lots of objects. Who thinks that's why it's hard? Who thinks that's not why it's hard? You think computers can kind of list a bunch of objects? It's easy, right? This is what I said about memory, right? It's a big, long list of stuff. Computers are good at that, right? There's going to be thousands of objects. A list of objects is not a hard thing for a machine to do. What's hard is that each object can produce an essentially infinite number of images. And so you somehow have to be able to kind of take some samples of certain views or poses of an object. This is a car under different poses, and be able to generalize or to predict what that car might look like in another view. And so really then you have to be able to, so you need some good, this is what's called the invariance problem. And it's due to the fact that, again, there's identity-preserving image variation. The car, this is why the barcode reader in your supermarket works fine, because the code is always laid out very simply. But when you have to be able to generalize across a bunch of conditions, potentially things like even background clutter, even more severely occlusion, things you heard from Gabriel, or you may even want to generalize across the class of cars, where the cars have slightly different geometry, but they're still cars. These kind of generalizations are what make the problem hard. So I'm lumping them all together in what we call the invariance problem. Okay, many of you in the room know this is the hard problem, and I think that hopefully that it fixes ideas, that's what you should think about. It's not the number of objects, but it's the fact that it has to deal with that invariance problem. Okay, this is Haim was talking about manifolds, and this is sort of my version of that. This is to introduce you to the problem of why that invariance problem, what it kind of looks like or feels like. I'm not going to give you math on how to solve it. It's just a geometric feel for the problem. So if you imagine you're a camera or your retina, which is capturing an image of an object, let's call this a person. I think I called him, here he is, Joe. So when you see this image of Joe, and this is the retina, so this is a state space of what's going on in your retina. So it's a million retinal ganglion cells. Think of them as being an analog value out of each. So this is a million-dimensional state space. So when you see this image of Joe, he activates every retinal ganglion cell, some a lot, some a little, but he's some point in that million-dimensional space. Okay, everybody with me? If everybody's heard all this before and wants me to go on, everybody raise your hand and I'll move on.
No, it's good.
Keep going. Okay. So the basic idea is that if Joe undergoes a transformation, like a change in pose, what that does is it's only a 1 degree of freedom. I'm turning under the hood one of those latent variables. If I had a graphics engine, I'm changing the pose of latent variables. There's only one knob that I'm turning, so to speak. And that means there's one line through here as Joe projects across these different images here. Now I'm ignoring noise and things. This is just the deterministic mapping onto the retinal ganglion cells. So Joe goes, and he goes over here. And if I turn the other knob, he goes over here. And so I could imagine if I turned those two knobs of two axes of pose all ways possible and plotted this in the million-dimensional state space, there'd be this curved up sheet of points, which you could think of Joe's identity manifold over those 2 degrees of view change. There's only 2 dimensions here. It's hard to start showing more than this, but it's sort of this curved up sheet of points. Everybody with me so far? You don't actually get to see all those, right? This is sort of, you could imagine a machine actually running them all, but you don't really get to see them. You're going to get samples of them, but there's some underlying manifold structure here. Now, what's interesting and what's important to point out is that this thing, even though I've drawn it, it's a little curved, but it's highly complicated in this native pixel space. It's all curved up and bending all over the place. And the reason that matters, and this is what Haim sort of introduced you to, is that if you want to be able to separate Joe from another object, say not Joe, another person, say, then you need a representation. So I showed you retinal ganglion cell. This is another imaginary state space where you can take simple tools to extract the information. And the simple tools that we like to use are linear classifiers, but you can use other simple tools. Haim used the exact same description to you guys in his talk. That you have some linear decoder on the state space that can say, oh, they can separate cleanly Joe from not Joe. So these manifolds are nicely separated by a separating hyperplane. That's what these tools tend to do, is they like to cut planes is one thing they like to do, or they want to find locations or regions, like compact regions in the space, depending on what kind of tool you use. But you don't want the tool having to do all kinds of complicated tracing through this space, or that's basically the original problem itself. So what you need is you have a simple toolbox, which we think of as downstream neurons. So a linear classifier is an approximation. It's like a dot product. It's a weighted sum, which is what we think neuroscientists of downstream neurons doing. And so it's a weighted sum. And so what we need is, like, if we want an explicit representation in some neural state space, then we need to be able to take weighted sums of some population representation to be able to separate Joe from not Joe and Sam from Jill and everything from everything else that we want to separate. We would call that, if we had such a space of neural population, we'd call that a good set of features or an explicit representation of object shape. And for the aficionados here, it's not just cleanly linear separation. It's actually being able to find this with a low number of training examples. So that turns out to be important. But it helps to just fix ideas to think about linear separation, ideally with a low number of training examples. So that's kind of a good representation. And when I notice I'm sort of starting to mix up terms here, I am assuming when I talk about shape that will map cleanly to identity, or what you might call broadly category. That's another topic I won't talk about. If you like, just think about the shape of Joe, or sort of separating one geometry from another. Okay, now here's a simulation that my first graduate student, Dave Cox, who's now at Harvard, did. This is a number of years old. This is take these same two face objects, render them under changes in view, and then he actually simulated the manifolds in a 14,000 dimensional space, and then he wanted to kind of visualize it, and because we wanted to just try to make the point that these manifolds of these two objects are highly curved and highly tangled, and this is a three-dimensional view. Remember, it's sitting in a 14,000 dimensional simulation space. You can't view that space. This is a three-dimensional view of it, and the point is that it's like two sheets of paper being all crumpled up together, and you can't. They're not fused. They look fused here because it's in 3 dimensions, but they're not actually fused. But they're complicated that you can't easily find the separating hyperplane to separate these two objects. So we call these tangled object manifolds. And really the job they're tangled due to image variation, right? Remember, if I didn't change those knobs of view or position or scale, there would just be 2 points in the space and it would be easy. That's the easy problem of listing objects. But if they have to undergo all this transformation, they become these complicated structures that need to be untangled from each other. So the way we think about what the problem that's being solved is you have this retina sampling data, like a camera on the front end, where things look complicated with respect to the latent variables, in this case, shape or identity, Sam or Joe. and that they somehow are transformed, as I mentioned, they're transformed by some nonlinear transformation, some other neural population state space, shown here, where the things look more like this. The latent variable structure is more explicit, that you can easily take things like separating hyperplanes to identify things like shape, which again roughly corresponds to identity, or other latent parameters like position and scale. You maybe haven't thrown away all these other latent parameters, and if I have time, I'll say something about that. So you don't just get identity, but if you can untangle this, you would have a very nice representation with regard to those original latent parameters. That's the sort of dream of what you'd like to do. It's sort of like reverse graphics, if you will. So this is what we call an untangled explicit object information, and we think it lives somewhere in the brain, at least to some degree, and I'll show you the evidence for that later on. So what you have then is you have a poor encoding basis, the pixel space, and somewhere in the brain is a powerful encoding basis, a good set of features. And as I mentioned, this, as I already said, this must be a nonlinear transformation, because the linear transformations are just rotations of that original space. So now let's go down to, actually, this would be MAR level 3. Let's go to instantiation. Like, let's get into the hardware here. We're supposed to be talking about brains. So I'm going to give you a tour of the ventral stream. So we would love to know how this brain solves it. This is the human brain. This is a non-human primate. This is not shown to scale, just blown up to show you it's a similar structure, temporal lobe, frontal lobes, occipital lobe. There's a non-human primate. We like this model because, for a number of reasons. One reason that we like it is that they're very visual creatures. Their acuity is very well matched to ours. In fact, even their object recognition abilities are actually quite similar to our own. This may be surprising to you, but let me just show you some data for that. This is actually data from Rishi Rajalingam in my lab. It says impressed with this, just came out. This is the confusion matrix patterns of humans trying to discriminate different objects under those transformations that I showed you earlier. Where they're not just seeing images, but they have to deal with these kind of invariances. And this is rhesus monkey data trying to do the same thing. And the task goes, I'll give you a test image, and then you get choice images. Was it a car or a dog? I'll show you an image. Choice image, was it a dog or a tree? And you're trying to entertain many objects all at once, and you get an image under some unpredictable view and unpredictable background, and then you have to make a choice. So this is the kind of confusion difficulty. And when you look at this, it's sort of intuitive that these are sort of geometry similar, right? Camel's confused with dog, and tank is confused with truck. And that's true of both monkeys and humans. And to some level, this shouldn't be surprising to you. The humans make the same tasks are difficult for humans are difficult for monkeys, because probably they share very similar processing structures. And this is not to bring, they don't have to bring in a bunch of knowledge about tanks are driven by people or that. They just have to say, was there a tank or a truck? And under those conditions, they make very similar patterns of confusion. And these patterns are very different from those that you get when you run classifiers on pixels or low-level visual simulations. But they're very similar to each other. In fact, are statistically indistinguishable, monkeys and humans, on these kind of patterns of confusion. Okay, so that's one reason we like this subject. The monkey model is that the behavior is very well matched to the humans. The other reason is that we know from a lot of previous work that I alluded to that some studies have shown that lesions in these parts of the brain can lead to deficits in recognition tasks. So again, we know that the ventral stream, or we think the ventral stream solves recognition, so we sort of know a weak word model of where to look. We just don't know exactly what's going on there. Just to orient you, these ventral areas, V1, V2, V4, and infratemporal cortex, or IT cortex, IT projects anatomically to the frontal lobe, to regions involved in decision and action, and around the bend to the mediotemporal lobe, to regions involved in formation of long-term memory. Because these are monkeys and not humans, and Gabriel mentioned this in his talk, we can go in and we can record from their brains, and we can perturb neural activity in their brains directly, and we can do that in a systematic way. This is the advantage of an animal model as opposed to a human model. Okay, as neuroscientists now, so now we've taken a problem, translated it to behavior, taken that behavior into a species we can study, and we know roughly where to look, and now we want to try to understand what's going on. So the ventral stream, as engineers, we take these curled up sheets of cortex and think of them, as I've already been showing you, as sort of populations of neurons. So there's millions of neurons in each of these sheets. I'll give you numbers on a slide coming up. There's some sort of processing that may be common. Here I put these T's, and there might be some common cortical algorithm processing forward this way. There's also intracortical processing, and there's also some feedback processing going on in here. So all that's schematically illustrated in this slide that I'll keep bringing up here when we talk about these different levels of the ventral stream. And I'm mostly going to be talking about IT cortex here at the end. Why do we call these different areas? One reason is that there's a complete retinotopic map, a map of the whole visual space in each of these different levels. In retina, there's one, and LGN, and in the thalamus, there's another, and V1 there's another map, and V2 there's another map, and V4 there's another map. In IT, there's it's less clear that it's retinotopic. We're not even sure that IT is one area. Maybe we'll have time. I'll say more about that detail. So it's not that retinotopic in IT, except the most posterior parts of IT. But that's why neuroscientists divide these into different areas. So the key concept, though, for you computationally is think of each of these as a population representation that's transforming the data from that complicated space to some nicer space. And it's doing this probably in a stepwise, gradual manner. So IT is believed to be that powerful encoding basis that I alluded to earlier, where you have these nice flattened object manifolds. And I'll sort of show you the evidence for that. Here's a more I guess this is recently from a review I did that gives more numbers on these things, and I've sized the areas according to their relative cortical area in the monkey. Here's V1, V2, V4, IT. IT is a complex of areas, and I'm showing you these kind of latencies. These are sort of the average latencies in these different visual areas. You can see it's about 50 milliseconds from when an image hits the retina to you get activity in V1, 60 in V2, 70. There's about a 10 millisecond step across these different areas. So it's about a 100 millisecond lag between when an image hits here and you start to see changes in activity at this level up here that I'm referring to IT. When I say IT, I'm referring to AIT and CIT together. That's my usage of the word IT for the aficionados in the room. And that's about 10 million output neurons in IT, just to fix numbers. In V1 here, you have like 37 million output neurons. There's about 200 million neurons in V1, similar in V2. You know, and many of you probably heard about other parts of the visual system. Here's MT. Many of you probably heard about MT. So it's sort of, you can see it's tiny compared to some of these areas that I'm talking about here. And I'm going to show you some neural data. I'm just going to give you a brief tour of these different areas. So brief, it's sort of almost cartoonish, but at least those of you who haven't seen this should at least be exposed. So in the retina, you guys know in the retina, there's a bunch of cell layers in the retina. The retina is a complicated device, right? I think it is a beautiful camera. So you're down in the retina. Now, to me, the key thing in the retina is the end, you've got some cells that are going to project back along the optic nerve, right? So these are the retinal ganglion cells. They actually live on the surface. The light comes through. Photoreceptors are here. There's processing in these intermediate layers. And then there's a bunch of retinal ganglion cell types. There's thought to be about 20 types or so. The original physiology, there's sort of two functional kind of central types where they have sort of on-center or off-center. Let's take an on-center cell. You shine light in the middle of the spot. Now this is a tiny little spot on the retina. The size depends on where you are in the visual field. But so you shine a little bit of light in the center, the response goes up. See the spike rate going up here. Put light in the surround, the response rate goes down. So it has an on-center, off-surround profile. And then there's a flip type here. So that's the basic functional type. When you think about the retina as it's tiled with all of these point detectors that have some nice center surround effects. There's some nice gain control for overall illumination conditions. But my toy model of the retina will be it's basically a real nice pixel map coming back down the retinal down the optic tract to the LGN. Okay, I'm going to skip the LGN and go straight to V1. People have known for a long time functionally V1 cells. They have sensitivity to especially edges. They have what's called orientation selectivity. Hopefully this isn't new to you guys. Here's a simple cell in V1. If you shine a bar of light on it inside its receptive field. Does everybody know what a receptive field is? I don't want to go. OK. It's OK if you ask, because I want to make sure you guys are OK. So the receptive field, you shine a bar of light in it, turn it on the right orientation, gets good response out of the cell, move it off this position. Now, not much response. There's a little bit of an off response here. Change the orientation, nothing happens. Full field illumination, nothing happens. Okay, so this is called selectivity. That is, there's some portion of the image space that it cares about. It doesn't just respond to any light at that spot like the pixel-wise the retinal ganglion cell would. So now there's this complex cell that's also in V1, which maintains this orientation selectivity across a change in position, as shown here, also across some changes in scale. But it maintains it, meaning that you have this tolerance, so that's called position tolerance for position. You can move the bar around, it still likes that oriented bar, but you change its angle and it goes down, right? So it still maintains the same selectivity here, but has some tolerance. So you get this buildup of some orientation sensitivity followed by some tolerance, right? And there are models from Hubel and Wiesel that are thought that you build this first and then you build these out of these. That's the simple version. And these here they are, these are the Hubel and Wiesel models, how you build these and-like operators to build selectivity from pixel-wise cells with an and-like operator lining these up correctly. You can imagine an oriented tuned cells built this way. There's evidence for this in physiology that this is how these are constructed. The tolerance of these complex cells is thought to build by a combination of simple cells, and there's some evidence for this. And this is, again, all the way from Hubel and Wiesel. They won a Nobel Prize for this and related work in the 1960s. And then there were a bunch of computational models that are sort of really inspired by this, and I think are still the core models of how the system works. And some of the original ones that were written down were Fukushima in the 80s, and then Tommy Poggio and others built what's called an HMAX model you guys have probably heard about, that's sort of off these similar ideas, much more refined and much more matched the neural data. And so, but these, I'm just trying to point out that these kind of physiological observations are what inspired this sort of class of largely feed-forward models. Again, you heard about much today. Okay, so that was sort of, that's what kind of a brief tour of V1. Now what's going on in V2? So for a long time, people thought, well, it was hard to tell the difference from V1 and V2. And I just thought I'd show you guys, this is a slide I stuck in. This is from Eero Simoncelli and Toni Mavshin. I think you guys have Eero teaching in the course a bit later, so he may say some of this. But V2 cells have some sensitivity to natural image statistics that V1 cells don't. And maybe I'll see if I can take you through this. So the way that they did this is you can simulate. So this is all driven off of work that Eero and Toni have done on, well, especially Eero has done on texture synthesis. So you have these original images, and if you run them through a bunch of V1-like filter banks, and then you kind of take a new image, a random seed, which is like white noise, and you try to make sure that it would activate populations of V1 cells in a similar way. There's a large set of images that would do that because you're just doing summary statistics. But these are some examples of them. For this image, this is one that one might look like. So you can see it doesn't look anything, to you it doesn't look the same as this, but to V1 these are metamers. They're similar in the summary statistics in V1. And then you start taking cross products of these V1 summary statistics, and then you try to match those. And what's interesting is you start to get something that looks texture-wise much more like this original image. And this is a big part of what Eero and others did in that work. And the reason I'm showing you this is that Toni's lab has gone and recorded in V1 and V2 with these kinds of stimuli. And the main observation they have is that V1 doesn't care whether you show it this or this. So these V1 to V1, these are both the same, which sort of says we have the summary statistics for V1 right in terms of the average V1 response. That's all I'm showing you here. The paper, if you want it, is much more detailed. But you go to V2, and there's a big difference between this, which V2 cells respond to more, and this, which they respond to less. And really, one inference you can just take from this is that V2 neurons apply a repeated another and-like operator on V1. That's a simple inference that these kind of data seem to support. And they also tell you that these and-like operators, these conjunctions of V1 statistics, tend to be in the direction of the statistics of the natural world. That's naturalistic statistics. Now, lots of controls haven't been done here to sort of narrow in exactly what kinds of ands, but that's the spirit of where the field is on trying to understand V2. Everybody thinks it has something to do with corners or a more complicated structure, but this is a way that current in the field to try to move these image computing models forward in V1 and V2. And Toni likes to point out that this is one of the strongest differences that you see between V1 and V2 other than the receptive field sizes. So I think that's sort of quite some exciting work if you don't know about it on V2. Okay, then you get up into V4 and things get much murkier. So what's going on in V4? Well, let me just sort of briefly say that one of my postdocs, this is more recent work, just because it builds on that earlier work. This is Nicole Rust, when she was a postdoc in the lab, compared V4. She actually compared it to IT. I'll skip that. But she was using these Simoncelli scrambled images. These are actually the texture images these are the original images, and these are the texture versions. So this should look like a textured version of that. You can see that these algorithms don't actually capture the object content of these images. And what Nicole actually showed is that V4 doesn't, similar to what you just saw there in the earlier work, like V1, V4 doesn't care about the differences between these. It responds similarly as a population to this and this and this and this and this and this. But IT cares a lot about this versus this. So this is just repeating the same theme. The general idea that you have and-like operators that we think are aligned along the ventral stream that are tuned to the kind of statistics that you tend to encounter in the world. And these are some of the evidence. This is some of the evidence for it in V2 and then later in V4 and IT and Nicole's work if you piece that all together. You go to a place like V4. Remember, V4 is now like 3 levels up. And what does V4 do? Look, this is Jack's work in 1996. So This is from Jack Gallant when he was working with David Van Essen. And people had some ideas that maybe there are these sort of certain functions that V4 neurons like, and they would show these sort of the same thing people have done in V2. They would show a bunch of images like this and sort of figure out, well, does it like these Cartesian gratings or these curved ones? And what you get out of this is, you can tell some story about it, but you get a bunch of responses out there. The color indicates the response. And then you kind of look at it, and people would tell some stories, but it really was just kind of like tea leaves. Here's a bunch of data. We don't really know what these V4 neurons were doing. This was a science paper, so you could go back and read it. Ed Connor and Anitha Pasupathy worked together a few years after that to try to sort of figure out more about what V4 neurons do. And they did things like take images like this, which were isolated, and try to cut them into parts, like curved parts, pointy parts, curved, concave, convex. And this was motivated off of some psychology literature. And they would define these based on sort of the center of the object. So this isn't an image computable model. It was just a basis set that they built around these kind of silhouette objects. And so they made this basis set about any kind of silhouetted object they like here. They hypothesized that they could fit the responses of V4 neurons in this basis set. And actually, this is their attempt to do it. They could actually fit quite well. And that's kind of what's being shown here. Here's the response of a V4 neuron. The color indicates the depth of the response. So you can see this is sort of like that previous slide. You're looking at tea leaves. It looks complicated. But under this model, they were able to, in the shape space, explain about half of the response variance of V4 neurons. So the upshot is that V4 curves about some curved curves or combination of curves. And then later, Scott Brincott with Ed went on into posterior IT and showed that maybe some combinations of these V4 cells could fit posterior IT responses quite well. So if you read the literature in V4 and IT, you'll come across these studies, and they're important ones to look at. Unfortunately, they don't give you sort of an image computable model of what these neurons are doing, but it's sort of some of the work that you should know about if you want to look in V4 or early IT. So I'm telling it to you. So let me go on to IT, which is what I want to talk about for the rest of today. Again, I'm talking about AIT and CIT. And let me just, I'll just quickly say that the anatomy again suggests that the IT is the central 10 degrees. And you can look at that based on, even though V1, V2, and V4 cover the whole visual field, if you make injections in V4, that's sort of shown here, where you make injections on the more peripheral parts of the V4 representation, which is sort of up here, that you don't get much projection into IT, which is here. You don't see much green color. Whereas you make projections in the center part of V4, these red sites here, you see much more coverage into IT, which is shown here. So it's, when I say 10 degrees, that's rough. Everything in biology is messy, but this is some of the evidence beyond recordings. There's anatomical evidence that as you go down into IT, you're more and more focused on the central 10 degrees. Okay, let me talk about a little bit of the history of IT recordings. This is what people got excited about IT in the 70s. This is work by Charlie Gross, who's one of the first people to record an IT cortex. And what they did was, I'll show you what they did here. This was in an era where, remember, Hubel and Wiesel had just done their work in the 60s, and they'd recorded from the cat visual cortex, and they had found these edge cells, and they've ended up winning the Nobel Prize for that. So it was sort of the heyday of like, let's record and figure out what makes cells go. So they were brave enough to put an electrode down in an IT cortex in 1970 and said, what makes this neuron go? Remember, that's an encoding question. What's the image content that will drive this neuron? And it's fun to just look back on this and what they were doing. So they didn't have computer monitors. They were actually sort of waving around stimuli in front of the animal. This is an anesthetized animal on a table. This was a monkey. Actually, they started with a cat, and then they later went to monkey. The use of these stimuli was begun one day when, having failed to drive a unit with any light stimulus, so that probably means spots of light, edges, things that Hubel and Wiesel had been using, we waved a hand at the stimulus screen, like waved in front of the monkey, and elicited a very vigorous response from the previously unresponsive neuron. Okay, so they said, well, look, and then we spent the next 12 hours. So the animal's anesthetized on the table. They're recording from this neuron. It's 12 hours because nothing's moving, so you can record for a long period of time. So single neuron, they're recording, listen to the spikes. We spent the next 12 hours testing various paper cutouts in an attempt to find the trigger feature. So that's you can see that's a Hubel and Wiesel idea. What makes this neuron go? What's the best thing? That's sort of become what a lot of the field spent time doing. Trigger feature for this unit. When the entire stimulus set we used were ranked according to the strength of the response that they produced, we could not find a simple physical dimension that correlated with this rank order. However, the rank order of adequate stimuli did correlate with similarity for us, that means psychophysically judged, to the shadow of a monkey hand. So they sort of, these are the rank order of the stimuli. And they say, well, look, it looks like it's some sort of hand neuron. That's all I know how to describe it. I can't find some simple thing on here. So this kind of study, then launched a whole domain where people started to go in and record these neurons, and they found interesting different types. Bob Desimone, who worked with Charlie Gross, later showed much more nicely under more controlled conditions. Yes, there are indeed neurons that respond. You can see more to these hands. This is the post-stimulus time histogram. Lots of spikes, lots of spikes, lots of spikes. Respond more to these hands than to these other kind of stimuli here. That is, so you could say these neurons are tuned to specific combinations of high selectivity. You'll hear from Winrich that others had shown that you can record some of the neurons that are really like faces that you could find and not so much hands, right? So you could find neurons that seem to have some interesting selectivity in IT cortex. And then others later went on to show in a number of studies, this is from Nico Logothetis' work a number of years later, it's just one example, that this selectivity had some tolerance to, say, the position of the stimulus. That's what's shown here. The fact that this bars are high just means that it tolerates movement in this is size, degrees of visual angle, and this is position, moving the stimulus around. So this was known for a number of years that there's some tolerance to position and size changes at least. Okay, so what I'm putting these up, you say there's some selectivity and there's some tolerance, and that should remind you of what we already said in V1. There's some selectivity, simple cells, there's some tolerance, complex cells, right? So you have the same themes here, just different kinds of types of stimuli being used. Then people really went on in the 80s especially and said, let's go after this trigger feature. And this Tanaka's group really went after this really hard. They tried to, Tanaka's group would find what's the best stimulus. They would dangle a bunch of objects in front of a recorded neuron, find the best out of a whole set of objects, and then they do reduction. They try to figure out how can I reduce this? This is their attempt to reduce the stimulus to its features without lowering the neural response. So high response, high response, high response, high response, high response. Suddenly I do this, the response drops. I do this, the response drops. And they have lots of examples of this. And they want you to sort of try to get to the simplest theme that could capture the response. And so when they did this, they would kind of take stimuli like this and end up with stimuli that looked like that. Okay? Now, many of you should probably start to kind of wonder here, there's lots of paths through stimulus space. So it's not clear that these are elemental in any way. There's lots of ways that you can show with modeling that you can kind of get easily lost in this space of navigating around here. This is just, again, a history of the work. This is the kind of things that people were doing. And then from that, they presented what we think of as the ice cube model of IT that I think is actually still a very reasonable approximation. They not only show that neurons tended to like certain these kind of relatively sort of reduced stimulus features, not full objects, but that they together. So these are millimeter scale regions of IT that nearby neurons within a millimeter or so have similar preferences. They're not just scattered willy-nilly throughout the tissue. When you go record nearby neurons, they're similar. So there's some mapping within IT cortex. This is schematic here. This is optical imaging data. Cortex, also from Tanaka's group, that show you that these different blobs of tissue get activated by different images shown here. And I'm just showing you the scale of this is around a little less than a millimeter, right? So, and our lab has evidence of this too. So there is some spatial organization in IT, but we really don't really yet understand features of these elemental features yet, or at least not at this time. Okay, so. Then later, there's lots of beautiful work in IT. Again, I'm probably not telling you all of it. Some of the most exciting work recently, and you'll hear about this from Winrich, that people started to use fMRI. So Doris Sau and Winrich Friedwald and Marge Livingston all together started to use fMRI to compare faces versus objects. This was motivated from human work by work like Nancy Kanwisher lab and others. That would then, what they found was that in monkeys you could find different parts that would show up what are called face patches, where you have a relative preference for faces over objects. Again, I don't want to take all of Winrich's talk here, but you have these different patches here. And then what's really cool is you go in and record from these patches, and that you find a very enriched locations for face neurons. And these enriched locations were known from a number of other studies, but this is a nice correlation between functional imaging and this enrichment of these face cells, and that's what's shown here, that these neurons respond to they respond mostly to faces and not so much to other objects, although you see they still sort of respond to these. So this kind of says fMRI and physiology are telling you similar things. It also tells you there's some spatial clumping, at least for face-like objects, at a scale of a few millimeters or so, the size of these patches. Okay, so that's larger scale organization. This is data from our own lab that sort of shows thing. Maybe I'll just skip through this in the interest of time, that we can map and record the neurons very precisely, map them spatially, and compare that with fMRI. So this is just a larger field of view maps of the same idea. Okay, so what we have then, just to kind of wrap up this whirlwind tour of the ventral stream, remember we have some untangled explicit information, and what I want to try to convince you of now is that I've told you about the ventral stream, but I'm going to try to tell you that in IT cortex this is a powerful representation for encoding object information. So the way, and then we'll take a break because we've already probably been going, well, a while. Yeah, about 10 more minutes, and then we'll take a break. So what I've told you is I've led you up the ventral stream. I've given you a bit of the history. So now let's talk about IT more precisely. So now this is work from my own lab. One of the things that you go in and record IT, you go record extracellularly, you travel down into IT cortex, which is down here, and you record from this. And similar to what you saw, modern version of what you saw from Charlie Gross or Bob Desimone, you show a bunch of images. And they could be arbitrary images. You take an IT recording site and see these little dots. Those are action potential spikes out of a particular IT site. And these are repeatable, right? You have some Poisson variability here, but you see that there's more spikes here, there's a little more here, less. These images are all randomly interleaved when you collect the data, as I'll show you in a minute. And you go to different sites and it likes different images, right? So there's certainly some image selectivity. This should not be surprising because I already showed you this from previous work. This is just data from our own lab. You can also see now that you're looking closely at this, the time lag around, remember I said around 100 milliseconds, stimulus on, stimulus off. The stimulus is actually off before the spikes actually start to occur out here in IT, because again, there's a long time lag, 100 milliseconds. Okay, so that's what the neural responses look like. Here, I think, I don't know if you guys can hear this. Maybe I should have hooked up audio. Maybe you might be able to hear. This is actually a recording that we did, that Chao Hung did when he collected this data in my early studies we did in the lab. I don't know if you guys can hear. Those high beeps are the animal getting reward for fixating on that dot. You're not even going to be able to parse that, right? I mean, you hear the spikes clicking by those are action potentials, right? And I don't expect you to look at anything like, oh, it's a face neuron or whatever. I just want you to get a feel for how those data were originally collected. This is pretty grainy video, but you get the kind of idea. And you collect data like that, and again, you can find selectivity in those population patterns, as I just showed you. But then Gabrielle and Tommy and I, so the three of us, I think, all in this room, way back when in 2005, said, well, look, the population of IT might have good, useful information for solving this difficult object manifold tangling problem. It might be a good explicit representation. So we did a sort of very first, what I call an early test of this idea. We took this sort of simple text from 8 different categories that we had chosen, and there's good stories of why we chose those objects if you like to hear them, but let me just say simple objects. We moved them across position and scale, and we collected the responses of IT of a bunch of sites to changes in to all these different visual images. And we showed them, as I just showed you, we just showed them for 100 milliseconds. This is this core recognition regime where we're just showing them for 100 milliseconds, and then we show another one, and they're just randomly interleaved. Okay, and from this, what you do is you can get a population set of data where we recorded 350 IT sites. Here's a sample of 63 sites. This is 78 images. The mean response here is the mean response to an image. This is 78 of the images we showed. There's nothing for you to read into here to say other than you have this rich population data. And now our question is, well, what is this data? What lives in this population data that we've collected? Is it explicit with regard to categories? So we come back to what I showed you earlier about those tangled manifolds and said, we need decoding tools. Let's say, kind of simple decoding tool. Look at that IT population and tell me what's out there. And again, we were using linear classifiers at the time because we took that, as you heard from Chaim, as our operational definition of what a simple tool is. And if it could decode information about the object identity, then we'd say, well, that means by that operational definition, this is explicit, available, accessible information, or just generally good. So if you imagine that the activity, this is schematic, each dot, this is neuron 1, neuron 2, and you could have a bunch of IT neurons. If you could separate any object from all the other objects, these points represent the population response to each image of an object. Remember, there's many images of each object. If you could linearly separate that, that would mean it was explicit. And if you had a hard time separating it, this would be implicit. These are like tangled object manifold. This is inaccessible or bad information. So when I mean we, I mean Chao Hung, who led the study, Gabriel, Tommy, and I did this. We took the response of an image like this one. It produced a population vector. Again, we recorded a bunch of neurons. We recorded them sequentially and then pieced together this population vector. And so these are the spikes simulated off a neuron, off of a population of IT. We could do various things, and in fact, I think Gabriel did everything possible, as I remember at the time. And one of the things we did was just count spikes. One of the simple things that turns to work quite well is count the spikes over 100 milliseconds. So this neuron counts spikes. That gives you a number, one number here. Count neuron spikes, get one number. So you have n neurons. You get n numbers. So it's a point in an n-dimensional state space, and where n is the number of neurons. And then we had already pre-divided the images into different categories, as shown here. These are the categories, and again, we just asked how well you could separate, how well you could do faces versus non-faces, toys versus non-toys, so on and so forth. These are old slides, but you get the idea is that basically you don't need that many sites to already get to very high levels of performance on both categorization and identification. The interesting thing about this was that you could solve this simple forms of this invariance problem in this representation quite easily. That if you just trained on the center objects, this center and size, a simple 3-degree size center position, and tested on the same thing, just held out repeats of this data, you did quite well. That's sort of a baseline. But what's interesting is you test in position and scale, and then you also do almost nearly as well. So you kind of naturally generalize to these other conditions by just training on these simple conditions. So this is evidence that the population is a good basis set for solving these kind of problems. A few number of training examples on this population, then generalizes well across these, make the problem hard. So again, we published that a long time ago. This was an early step to say, look, the phenomenology looks right for the story that I've been telling you so far. You can't do this easily in earlier visual areas like V1 or simulated V1 or V4, and we later showed that a number of ways. This is consistent with showing you logos-theta's position tolerance, size tolerance, the selectivity. It's really just an explicit test of the idea of population encoding. So the take home here is that there's this explicit object representation in IT. I didn't prove to you that this is the link, this predictive model, to decoding yet. We're going to talk about that next, but this was some of the important population phenomenology that we did. What I try to tell you today, hopefully I've introduced you to the problem of visual object recognition and the way we restricted it to core object recognition. We talked a lot about predictive models as being the goal, although I haven't presented much to you yet. Hopefully that's the second part of the talk. I've given you a tour of the ventral stream, but I mean, it was a poor tour. I'm sure everybody that works would say that you've neglected all this work, because there's no way I can do that all in even a whole week. I just try to hit some of the highlights for you. And I told you that the IT population seems to have solved a key problem, this sort of invariance problem that I set up. And one way to step back and say, over the last 40 years or so, from those Charlie Gross or even Hubel and Wiesel, the field of ventral stream physiology, we've largely described important phenomenology. Even that last study is sort of population phenomenology. And so now we need these sort of more advanced models. And so the next phase of the field is developing and testing these predictive models that I motivated at the beginning, but I haven't given you much of yet. So this was hopefully a bit of history and sort of set context to where we are.